Good afternoon. My name is Jessica Schofield, and I'm an assistant professor at the University of Alabama at Birmingham. I would like to first thank the organizers of this symposium for the invitation to talk about our research today. And today I'm going to discuss how commensal streptococci interfere with Pseudomonas aeruginosa persistence. First, I would like to state that there are no relationships that I need to disclose for this presentation. So Pseudomonas aeruginosa is the main pathogen that we work with in the lab. It is a gram-negative opportunistic pathogen and is distributed in most environments. It's a soil bacterium, but it has the ability to live in any environment. It is a pathogen of multiple hosts, so it can infect plants, fungi, nematodes, and insects, as well as humans, of course. And one of the reasons why it is such a successful pathogen is because it has a large repertoire of genes devoted to virulence, metabolism, and transport. And it also has the ability to alter the regulation of virulence genes to adapt to different environments. It is multi-drug resistant, and it forms recalcitrant biofilms that are resistant to not only the host immune response, but antimicrobials. And it is well known for being a major contributor of morbidity and mortality in cystic fibrosis patients. Now, typically in CF, these patients are colonized in the lung with multiple species of bacteria. But as you see here, Pseudomonas in the blue line tends to become dominant in the CF lung over time. And the majority of patients with CF are colonized with Pseudomonas aeruginosa. However, recent evidence or recent published studies suggest that there are other species of bacteria that are colonized within a lung. And so this is data from work by Filkins et al. That came out in 2012. And they sampled CF sputum from inpatient unstable population and outpatient patients that were clinically stable. And as you see in the red, which is Pseudomonas, Pseudomonas is dominant in a lot of the patients that you see. However, Streptococcus was prevalent in the outpatient clinically stable patients, as you can see in the green. And what was interesting is that this study showed that the colonization of oral streptococci in the lung was actually associated with improved lung function. As it states here, these patients were clinically stable. And Streptococcus parasanguinus was the second most abundant streptococcal species found in these stable patients. However, the mechanism in which streptococci used to improve CF lung function is currently unknown. And so Streptococcus parasanguinus is an oral commensal, and so it is an early colonizer of the tooth surface. It's normally acquired shortly after birth, and it's predominant on the tongue. And it produces hydrogen peroxide. It is alpha hemolytic, and it's been associated with oral and systemic health. However, how this particular commensal contributes to improved lung function is currently unknown. And so we've hypothesized that Strep parasanguinus interferes with Pseudomonas pathogenesis, particularly because we know that Pseudomonas is a major pathogen in the CF lung, and so we believe that parasanguinus might somehow interfere with Pseudomonas aeruginosa virulence. And so the approach that we take to study Pseudomonas aeruginosa and Streptococcus parasanguinus interactions is to use in vitro and in vivo models to study how they impact one another. So throughout this talk, the commensal that I'm going to talk about is Strep parasanguinus, and you would see it denoted as SP. And I'm also going to talk about two different strains of Pseudomonas aeruginosa. The first is called PAO1, which is an acute wound isolate, and the second is FERDA1, which is a chronic CF isolate. So the first thing we did to see how these two species interact with each other is to do a plate inhibition assay. And to do this assay, we spot the bacteria side by side and we look for any inhibition by one species towards the other. And so here, on the left, we have the Pseudomonas, and this is the PA01 strain. This is the Furta1 strain, which is the chronic CF isolate. And on the right, we have the commensal Strep parasanguinus. Now, initially, when we started these studies, we grew these anaerobically, and because of the anaerobic growth, we had to supplement with nitrite because Pseudomonas requires that for anaerobic growth. And so we saw in the presence of nitrite that Strep parasanguinus was actually inhibiting both the acute isolate PA01 as well as the wound or the CF isolate Furta1. And when we co-culture these together in liquid culture, we got similar results. And so these are CFUs of PA01 when it's grown by itself plus or minus nitrite, and then when you add Strep parasanguinus. And so you see a reduction in PA01 viability when nitrite is supplemented in the media. Similar story for Furta1, but what was interesting about this particular piece of data is that Furta1 is a lot more sensitive to the effects of nitrite and parasanguinus compared to PA01, the acute isolate. And so we wanted to know exactly how does parasanguinus interfere with Pseudomonas aeruginosa viability in a nitrite-dependent manner. And as I mentioned before, hydrogen peroxide is a major virulence determinant produced by Strep parasanguinus. And so we questioned whether hydrogen peroxide had anything to do with the phenotype. So we knocked out the gene required for hydrogen peroxide by Strep parasanguinus. This gene is called POXL, or pyruvate oxidase. And so when we grew the POXL mutant in the presence of nitrite, so this media has nitrite, and so when we grow the POXL mutant next to Pseudomonas, it abolishes the inhibition of Pseudomonas. And so on the right, we have parasanguinus, and on the left, we have Pseudomonas for the PA1 isolate and for the Furta1 isolate. So again, hydrogen peroxide is required for inhibition of Pseudomonas. And of course, when we complemented the POXL gene, it restored inhibition of Pseudomonas. And so we wanted to know what was the potential mechanism of inhibition of Pseudomonas by hydrogen peroxide from parasanguinus and exogenous nitrite. And so we know that both hydrogen peroxide and nitrite provide precursors for a reactive nitrogen species called peroxynitrite. And so we wanted to know whether or not peroxynitrite was actually involved in the inhibition of Pseudomonas, in addition to whether POXL mediated production of peroxynitrite by the commensal Strep parasanguinus. And so here we measure peroxynitrite in wild-type Strep parasanguinus in the POXL mutant and the POXL complemented strain in cultures that had no nitrite and cultures that had 1 millimolar nitrite. So as you can see, the addition of 1 millimolar nitrite dramatically increased the production of peroxynitrite. And as I mentioned before, peroxynitrite is a reactive nitrogen species that has potent antimicrobial activity. And what's interesting about this piece of data or this finding is that higher peroxynitrite concentrations have been correlated with improved lung function in CF patients, which suggests that peroxynitrite is indeed found or being made in the CF lung, and it potentially has some antimicrobial activity against CF pathogens. And so we wanted to know exactly how the addition of Strep parasanguinus and nitrite was impacting the transcriptome of both the PA1 and the Furta1 Pseudomonas isolates. And the take-home message, and these are genes that are involved in reactive nitrogen stress or reducing nitrate to nitrite. So these are denitrification genes. What we saw is that when you expose parasanguinous and nitrite, or expose PA1 and FERD1 to parasanguinous and nitrite, these two isolates respond very differently. And so most of the genes were turned on in the PA1 background, but FERD1 was unable to highly induce the expression of these genes when it was grown with parasanguinous and nitrite, which suggests that parasanguinous might dysregulate the ability of a chronic CF isolate to properly respond to reactive nitrogenous stress. And so because these genes are involved in responding to reactive nitrogen species, we question whether mutations in these genes, particularly nitrite reductase, which reduces nitrite to nitric oxide, as well as the nitric oxide reductase, were they involved in the ability of Pseudomonas to respond or to resist or become susceptible to parasanguinous mediated nitrosity stress. And so we created mutations in nitrite reductase, the NERS gene, in both the PA1 and FERD1 background. And interestingly, we saw that loss of nitrite reductase made Pseudomonas more susceptible to parasanguinous even without the addition of nitrite. And of course, this was restored. The growth was restored in the complemented strain. And so next we tested whether two different nitric oxide reductase genes had any involvement. So we tested mutations or mutants in NOR-B and NOR-C in the PA1 background. And so in our control media, where we don't have any nitrite, we didn't see any inhibition. However, in the NOR-B mutant, when it was exposed to parasanguinous and nitrite, it was more susceptible to parasanguinous and nitrite-mediated stress compared to wild-type PA1. Similarly, we had increased killing in the NOR-C mutant compared to wild-type PA1, which suggests that NOR-B and NOR-C are critical for the response to reactive nitrosity stress by strep parasanguinus. And so far we know that at least in our in vitro models that strep parasanguinus and nitrite can inhibit Pseudomonas growth for both an acute isolate and the cystic fibrosis isolate FERD1. But we wanted to determine whether or not this translated into protection in an in vivo model. And so we used Drosophila as an in vivo model. And one reason why we use this model is because it is a quick infection model just to determine whether or not Pseudomonas has attenuated pathogenesis. We also know that the same virulence determinants that Pseudomonas uses to infect humans, it also uses to infect Drosophila. And so we knew that we could use this model to test whether or not parasanguinus and nitrite offer any protection. And so when you have single and dual infections of parasanguinus and Pseudomonas, in addition to parasanguinus that is deficient in pyruvate oxidase, so meaning that it does not produce hydrogen peroxide, you do not see any difference in the killing of Drosophila over the 8-day period. And this is, of course, is without nitrite. So there's no difference between Drosophila survival in the co-infection model when you have Pseudomonas by itself and we're using PA1, or you have Pseudomonas with parasanguinus or Pseudomonas with the poxyl deficient mutant. But what is interesting is that once you add nitrite to the system, you get a huge increase in Drosophila survival. So in the presence of strep parasanguinus wild type, you we got 80% survival of these Drosophila flies compared to flies that were singly infected with Pseudomonas. And the pox cell deficient mutant had marginal level of protection. So these data suggest that Strep parasanguinus and nitrite may offer protection against Pseudomonas infection. So taken together, our working model suggests that the presence of hydrogen peroxide producing oral streptococci in the presence of nitrite can produce an antimicrobial called peroxynitrite. And we believe that we know that peroxynitrite has antimicrobial activity against Pseudomonas and can result in Pseudomonas cell death. And so we believe that this might potentially be involved in improved lung function in cystic fibrosis patients. So to conclude, the oral commensal Strep parasanguinus inhibits Pseudomonas in a hydrogen peroxide and nitrite dependent manner in both our in vitro and our in vivo models. We know that the chronic CF isolate FER1 is more susceptible to Strep parasanguinus mediated stress, and we believe that this increased susceptibility might be due to the inability of FER1 to properly respond to nitrosative stress. And we know that Pseudomonas denitrification genes are critical for survival during co-culture with Strep parasanguinus. So going forward, we think that promoting commensal streptococci prevalence in the CF lung might be useful for attenuating Pseudomonas pathogenesis. And so our future directions are to determine whether Strep parasanguinus and nitrite provide protection against Pseudomonas in a CF rat model. And we also want to know how Strep parasanguinus and nitrite modulate the host immune response. There are no studies currently that look at how Strep parasanguinus impacts the host immune response in addition to nitrite. And so currently we have developed a co-infection model of Pseudomonas and Strep parasanguinus using a CF rat model to determine, can it provide protection? Can parasanguinus and nitrite provide protection against Pseudomonas in this model? And how does that impact the inflammatory response? So with that, I would like to thank my lab members. Joshua Beatty did a lot of the work with the NORB, NORC mutants, and Joshua Huffines was involved in gathering the RNA-seq data. Thank you.
Hi, I'm Vaughn Cooper, an evolutionary biologist and microbiologist at the University of Pittsburgh School of Medicine. And I'll be speaking about evolved adaptations by gram-negative opportunistic pathogens in the cystic fibrosis airway. I want to thank the organizers of this session for the opportunity to speak. I have the following disclosure of being involved with the establishment of the Microbial Genome Sequencing Center. Please share comments and questions throughout this talk. I want to encourage you to submit questions for the live panel discussion, and I'll also be monitoring the chat. Feel free to connect with me on the platform or via Twitter. So it's important that we recognize that most cystic fibrosis pathogens are opportunists and not human adapted. Rather, they're natural inhabitants of the environment. And this is true for the major gram-negative pathogens that we see rising in prevalence as cystic fibrosis patients age. And these include organisms like Pseudomonas aeruginosa, Cenotropomonas, Achromobacter, and the Burkholderia cepacia complex. These are the organisms that I'll be addressing today. These organisms live primarily associated with crop plants, in soils and waterways, and even in close association, even in symbiotic relationships with nematodes and insects. It's also true that they're increasingly being found as sort of natural inhabitants of our engineered environment, particularly in our water systems. These are complex, metabolically diverse, heterogeneous, fluctuating environments. And so these microbes have adapted to live in a variety of situations with diverse metabolic capacities. Now, the cystic fibrosis airway exerts some unique selective pressures. These include an acidic mucus, which also generates anoxic conditions as the microbes become buried in that. Microbes often attach to our airways, our cystic fibrosis airways in aggregates and form biofilm, and those conditions are favored. The cystic fibrosis airway is obviously treated periodically with antibiotics, especially aminoglycosides like tobramycin. The CF airway is certainly defended by our immune system, including phagocytes, complement adaptive immunity, and other elements from our adaptive immune system. And the nutritional conditions are distinct, comprised often of glucose, lactate, and amino acids as primary nutrients. And so I want to offer the hypothesis that none of these forces are entirely novel to these microbes on their own, and otherwise they would not colonize, but they are novel in their combination in the CF airway and in their relative constancy. So these conditions may be seen periodically in the natural environment, but they're seen relatively constantly in the CF airway. So these microbes are diverse. Each strain differs considerably in their ability to grow in a variety of substrates. And let's just consider that we have a great deal of variation across environments within these species. And some strains are more fit than others. So if we consider the biofilm life cycle, which involves attachment, forming aggregates, forming maybe more elaborate biofilms, and then ultimately dispersal and reattachment, some strains are better at sticking, and whereas others are better perhaps at a more modal or swimming environment. And that's represented in this figure with the y-axis fitness. Now, when microbes are exposed to extremes of these environments, genetic variation within those species is selected, including new mutations, and that those mutations might increase or broaden the niche breadth, maybe making some strains better at sticking and some strains better at swimming over here. If we can consider more specifically the CF environment, that also might involve selection for growth in low oxygen, growth under high antibiotics. And the reason why these microbes can colonize is that there's a decent number of individuals within these microbial populations that have reasonable fitness under this combination of environments. And then selection tends to act over here to augment and improve fitness under the extremes of these conditions. So selection is acting on naturally occurring new mutations arising in these infections to improve fitness in a combination of these conditions. So what are those sorts of adaptations? Well, if we step back and think about how we recognize adaptations in all cases, a hallmark of adaptation is parallelism or convergent evolution. And so if we consider these desert adapted creatures, you'll note that they've all gained the ability to blend in with their natural sandy environment by evolving colors, a sort of sandy colored exterior. And likewise, these two other desert adapted creatures have large ears, which helps them dissipate heat loss, dissipate heat and tolerate the high temperatures. These are clearly adaptations to the desert environment. We can take the same approach using whole genome sequencing of microbes sequentially acquired from chronic infections to ask, do we see mutations arising in parallel in the same gene or gene systems from bacterial isolates from chronic infections? These mutations then predict adaptations that we can test in the lab. And today I'll be focusing primarily on two traits: the ability to resist the commonly used antibiotic tobramycin, and the ability to grow under anoxic conditions and also within macrophages. So the first part is recently published research led by PhD student Michelle Scribner, Shelley from my lab, Alfonso Santos Lopez, and Chris Marshall, postdocs from my lab. Chris has since moved on to an independent position at Marquette, and Chris Dietrich, a bioinformaticist from my lab. Tobramycin is one of the most commonly prescribed antibiotics in the chronic airway conditions. In fact, the microbes are sort of bathed in a periodic dosage of tobramycin. It's aminoglycoside that disrupts translation, and what it typically does is bind and interfere with translation and prevents growth and leads to death. So microbes can evolve or have inherent resistance mechanisms to tobramycin by mutations that affect the target site where tobramycin binds the ribosome, by enzymes that modify the antibiotic, and also by proteins or enzymes, exterior surfaces that decrease drug accumulation at the exterior. Unfortunately, the genetic causes of resistance remain often unknown. So to help us narrow our genetic spectrum, we conducted some evolution experiments with Pseudomonas aeruginosa and also another gram-negative pathogen, Acinetobacter baumannii, to identify shared or parallel adaptations to tobramycin. And what we did was we sequentially increased the concentration of the drug from 0.5 times the minimum inhibitory concentration, MIC, to 4 times the MIC by day 12. And we evolved under 2 regimes, sort of planktonic serial transfer, and also in a biofilm environment where bacteria are selected to attach to a plastic or polystyrene bead in test tubes. And what we found was quite surprising to us, and that was that the 2 genes really leapt to the top across both species and in both environments. Those 2 genes, the elongation factor G, encoded by FuseA1, and a phosphoenolpyruvate phosphotransferase, PTSP, specific to nitrogen, were selected in nearly all populations in all environments. And neither of these are known targets of the drug, and neither were previously reported as clear causes of resistance to aminoglycosides like tobramycin. So the fact that these 2 leapt as sort of the early drivers of, likely drivers of resistance was a surprise. Again, although elongation factor G is key to translation, it is not known to be a drug target of tobramycin. So we also found mutations that are lifestyle specific. So these are those that affect LPS biosynthesis for Pseudomonas, and those that affect the electron transport chain, and also likely the exterior in Acinetobacter. We were able to define genotypes of Pseudomonas aeruginosa FuseA1 in combination with mutations in ORF-N, which encodes the LPS biosynthesis, and PTSP. And we found that these mutations in combination produced additive increases in resistance to tobramycin, as well as other aminoglycoside. And over here you can see these are the resistance levels, increased resistance levels to tobramycin. These also confer resistance to amikacin, gentamicin, and confer low-level resistance to tigecycline. We found these mutations are common in clinical isolates of Pseudomonas aeruginosa, but also, as I'll show, in Burkholderia, Cenocepacia, Stenotrophomonas, and Achromobacter. So here is a summary of surveys of isolates from the CF airway. And we see an abundance of mutations in FuseA1 previously unrecognized. Also some mutations in PTSP. And we also, more recently, and not yet published work, discovered an array of mutations in FuseA1 during chronic infections of Burkholderia cenocepacia in the CF airway in collaboration with the Lupuma lab, excuse me. We also then, I also recently analyzed recent genomes from Achromobacter and Stenotrophomonas in studies from the Marvig lab and from Gruschel et al. From this year. And again, we find mutations in FuseA1 that would be consistent with increased resistance to aminoglycosides. Now, these mutations cause aminoglycoside resistance in various species beyond the ones that I've shown. So we can show here, these are surveys of mutations that from studies of resistance to an array of aminoglycosides, and they're found in clinical isolates. And these mutations affect, they're clustered in certain regions of elongation factor G, but they're not sort of unique to a particular domain, suggesting this is a general diminishment of function of elongation or alteration of function of this factor that promotes aminoglycoside resistance. And I also wanted to point out that we found plenty of evidence of mutations in these ORF genes in Pseudomonas from these clinical isolates. And finally, I want to point out that we've seen that these mutations, elongation and fuse A in Burkholderia, contribute to these long-lived persistent infections in the CF airway. And so this is one that's sort of arose early and led to a long-lived lineage during a chronic B. Sanocypatia infection. Okay, so I've shown that you can use whole genome sequencing to identify parallel mutations affecting the same gene in bacterial isolates. What about fitness under anoxia and within macrophages? So here I'll turn to another study that we completed a few years ago in collaboration with Inês Silva and Leonela Moreira from Portugal. This is one patient that's been chronically infected with Burkholderia multivarians for over 20 years. We published this in M Systems in 2016. And so while this population underwent this period of steady diversification, which has been consistent with a lot of studies of adaptation to the CF airway by Pseudomonas and other strains, other species, what we found is that we did a phylogenetic analysis. This forms multiple co-occurring lineages that coexisted during this population, and thus each of these forms distinct clades, which can be viewed as a sort of a semi-independent evolutionary trajectory. So we can ask the question, do we see parallelism even within the infection? And we actually did. We found recurrent evolution of multiple mutations, including this gene set, the FIX gene, FIX L and J. And what's really remarkable about FIX L and J is that it also was one of the most commonly mutated genes in a really elegant series of studies of Burkholderia dolosa infections, another Burkholderia cepacia complex species, led by Tammy Lieberman with Greg Preby and Roy Kishoni in Boston. These were among the most frequent evolved during Burkholderia dolosa infections. Now, more recently, in collaboration with the Preby lab, Matt Schaefer's and Chris Marshall from my lab have discovered that these mutations really target the sensor domain in FIX-L. And when these mutations arise within patients and rise to sort of detectable frequencies, they're associated with worse outcomes. So here I'm showing results from the Preby lab that patients without FIX mutations undergo a relatively steady and sort of predictable decline in lung function with age. But when they acquire mutations in FIX, they're associated with more rapid loss of FEV after those mutations arise. So that motivates the hypothesis that maybe these increase virulence. We then step back and ask, do we see these mutations in other Burkholderia cepacia complex species? And in fact, in collaboration with multiple groups around the world, we see mutations in FIX during chronic CF airway infections by Burkholderia sanocypacia. We see that in the United States and in Portugal, Burkholderia contaminans in Portugal and in Argentina, and Burkholderia multivarians in British Columbia and in the United States. So to test the hypothesis, these actually are associated with virulence. Matt Schaefer's engineered these mutations into a common genetic background of clinical isolates and found for two species, dolosa and multivarians, that the evolved mutations were associated with increased fitness in macrophage and also an array of increased virulence. So what you see here are these asterisks, effects of engineered mutations in dolosa and multivarians, and they denote significant advantages in fitness. So here again, we can use whole genome sequencing to identify parallel mutations in the same gene. These mutations are associated with worse outcomes in Burkholderia cepacia complex infections. Now, I just want to quickly say that anyone who's been watching this seminar has likely been thinking, well, know that organisms, gram-negative pathogens, adapt to the CF airway. And we know that there are these traits that we see often, those involving, say, loss of motility, changes in central metabolism, dysregulation of quorum sensing, like mutations in LAS-R, in Pseudomonas aeruginosa, alterations in LPS biosynthesis, which long have been thought to be associated with evasion of host defense, but we actually also show, in conjunction with the tobramycin study, these actually directly increase biofilm production and antimicrobial resistance. And here, I'll show you yet another bit of data to support this. These are repeatedly observed mutations in chronic B. cenocepacia infections from sets of sibling pairs. We see mutations affecting flagella and thus reducing motility, and mutations affecting central carbon metabolism, iron uptake, and EPS biosynthesis. These are among the more common. This is frequency with a histogram. So among the genes that are hit most common tend to cluster in these groups. So bacteria evolving in new environments like the CF airway under strong selection experience stresses, and what they wind up having to do is evolve quickly. That is, as Zuckerberg said about Facebook, they have to move fast and break things. Except that natural selection has the ability to winnow among all of the broken things, newly arising mutations, and select out the best mutations. And so we can conclude that colonizing the CF airway produces very strong selection for short-sighted solutions that are locally adaptive to the CF airway. They'd likely be maladaptive in the natural environment. We wouldn't expect to see them in nature, particularly because they tend to affect major regulators with major side effects, generating trade-offs. But in the CF airway, they are potentially repeatable and thus predictable, which I'd argue that we could use to guide diagnostic and treatment strategies, asking questions like how adapted is this microbe and what drugs might be effective. So I want to thank you for joining in and for your attention. I want to thank my lab, particularly fans of these people, of these microbes in my lab, like Alfonso, Chris, Katrina, and Shelley Scribner, who's not pictured. And I want to thank my funders.
Hi, everybody. My name is Peter Belenke, and I'm an assistant professor in the Department of Molecular Microbiology and Immunology at Brown University. And I'd like to thank the organizers for the opportunity for me to tell you about our work that relates how host and microbial metabolism impacts the susceptibility of bacteria in the microbiome. And in many ways, the work in my lab is driven by two interrelated factors. There's a growing rate of antimicrobial resistant infections. And in 2019, according to the CDC, there were approximately 3 million resistant infections, and they led to about 35,000 deaths nationwide. There's also a growing number of superinfections that are caused by antibiotic-induced microbiome disruption, things like C. diff. And so both of these factors, antimicrobial-induced disruption and higher levels of resistance, drive our need to develop new ways, more efficient ways to treat infections. And in 2013, the CDC put out several ways that we could improve antimicrobial therapy, including prevention of ongoing infections, developing new diagnostic tests, promoting the development of new antibiotics. But the other thing they talked about is repurposing and improving the use of our large arsenal of available antibiotics. And that's really what my work has been focused on. And in order to do that, we need to have a complete understanding of how antibiotics kill bacteria in vitro, but also in the context of the host. And so a lot of in vitro work from me and many others have demonstrated that an important determinant of antimicrobial susceptibility in the host in vitro is is metabolic activity during antibiotic exposure. And in general, bacteria with repressed metabolism, let's say bacteria in a biofilm or maybe TB that has ways to shut down various aspects of metabolism. So bacteria in which metabolism is less efficient tend to be tolerant to antibiotics, whereas bacteria with high metabolic activity tend to be susceptible. So a way to think about this is that essentially the availability of metabolites and things like oxygen is a way to tune the susceptibility of a bacteria, at least in vitro. So another way of saying that is that in general, active metabolism, the ability to convert glucose to ATP rapidly, contributes to susceptibility, whereas less active metabolism, so a slower rate of glucose to ATP conversion, tends to be associated with tolerance to antibiotics. And this is a very important factor and something that we are beginning to really understand in the context of a bacteria in the test tube. The question I really want to know is, how does this impact susceptibility of bacteria, both in localized infections, but also importantly in the context of the microbiome? And you can see that you can have vastly different metabolic environments that a bacteria can encounter when it's being exposed to an antibiotic in that context. A lot of work has already looked at how antibiotics impact the microbiome, and we know that after during antibiotic administration, there's a dramatic change in the metabolite in the bacteria that are found in the microbiome. So antibiotics cause extinctions and disruptions and cause most bacteria to disappear, some bacteria to bloom. Essentially, really dramatic rearrangement of the composition. And this is associated with many acute consequences of antibiotics. So many of us have probably encountered some gastrointestinal discomfort, but also things like C. diff, fungal infections, and other superinfections are considered kind of the acute consequences of antibiotic therapy. Luckily, in most cases, our microbiomes are highly resilient and recover quite rapidly back to baseline within days, months, but definitely within years. However, in some patients that have prolonged antimicrobial exposure, or let's say like a CF patient where you have multiple rounds of antibiotics that are given throughout the year, the lifetime, there are long-term consequences from antibiotic-induced disappearances and dysbiosis, things like obesity, IBD, inflammatory conditions, diabetes, and many others. Thus, the conjecture is that as we work to improve antimicrobial therapy, we need to be cognizant of the impacts on the microbiome and find ways to protect this community. And that is probably really important in CF patients as well. So this is a paper where they looked at a group of healthy adolescents in green, and CF patients that are obviously given multiple rounds of antibiotics in red. And as you can see, as time goes on, the diversity in their microbiome significantly deviates from the healthy cohort. So they're much less diverse, likely due to multiple rounds of antibiotic therapy, but also probably due to physiological differences in these patients. So the question that I'm really interested in asking in my lab is why, when you give an antibiotic, do some bacteria survive and some bacteria don't? And my conjecture is the metabolic activity is going to play an important role in that. To answer this question, I worked with a student named Damien Cabral, and I'm sorry that my picture is apparently blocking his picture right now. But Damien did a really cool project. We essentially gave mice 3 different types of antibiotics for a really short period of time, just to see what the initial impacts are. Then we extracted the DNA and the RNA. We used the DNA for 16S, but also shotgun metagenomics to find out essentially who is there. And then we used the RNA for shotgun metatranscriptomics to find out the transcription responses of the microbiome as a whole and for individual bacteria. So essentially, we can find out, who survives and who dies during antibiotic exposure, and what are the transcriptional activities associated with either death or survival. And then once we do that, we can then change the dietary inputs to figure out how that then impacts susceptibility. And so this is some of the data from the experiment. And so these are the top 50 bacterial species as detected by shotgun metagenomics. And you can see that each antibiotic has very dramatic impact on the composition. So red or yellow is kind of up and blue is down. And you can see that essentially doxycycline and ciprofloxacin have relatively limited impacts on the microbiome, whereas in this case, at this concentration, amoxicillin really wipes out essentially all of the bacteria except for 4. And those 4 are a lot of those 4 are in the Bacteroides phylum, and one of them is a bacteria called B theta. So B theta is an important member of both the human and the murine microbiome, and it seems to be bloom during antimicrobial therapy. It goes from about 30% to close to 90% when we give these mice amoxicillin. Yet when we give doxycycline or ciprofloxacin, it actually seems to die. Interestingly, if you look at the susceptibility of this bacteria to amoxicillin, it should be kind of relatively susceptible. It's probably not the best drug to use to get rid of this bacteria, but theoretically it should be susceptible. So then we looked at the metatranscriptomic analysis of the microbiome as a whole. In this case, we utilized human 2 to do this analysis. And let's just look at the amoxicillin data because that's kind of what I'll focus on for the rest of the talk. And we found that the resulting microbiome after amoxicillin exposure seemed to shut down a lot of metabolic products compared to the untreated mice. And specifically, things responsible for really rapid energy generation, parts of the TCA cycle and glycolysis were very much knocked down in these mice. So it appears that the surviving microbiome seems to be less metabolically efficient than the non-treated microbiome. And this aligns with the overall hypothesis that if you're metabolically deficient, you're more likely to survive exposure. We then looked at the specific enzymes responsible for fiber degradation. And we found that the treatment also, amoxicillin treatment in this case, also had very dramatic impact on the types of fiber that could be utilized by the microbiome. Amoxicillin was associated with more processes that were breaking down alpha bonds, and the breakdown of more host-derived polysaccharides versus dietary polysaccharides. And then the production and the breakdown of more kind of storage polysaccharides as well. So very dramatic change in the types of fiber that the microbiome is able to utilize. Next, we did this really cool experiment, probably my favorite experiment. I'm so glad that Damien suggested that we do it, is we gave mice amoxicillin for a day. We took their cecal microbiome, extracted it, and then put that on these biolog plates. So essentially, there's a 96-well plates that have different carbon sources, mostly simple sugars, so amino acids, things like that. And we found that if we compare the microbiome of an untreated mouse in blue and the microbiome of a treated mouse in red with amoxicillin, that on these biolog plates, the utilization of these simple carbohydrates is much less efficient in the treated microbiome. This is not explained by the overall number of bacteria that we're putting into each well, because interestingly, amoxicillin doesn't reduce the overall number of bacteria, it just changes the composition. Next, we utilized our shotgun metatranscriptomic data to do species level analysis. And essentially what we can do is we can take our reads, and for abundant bacteria like B theta and other members of the microbiome, we can then bin all of the reads that we get from that bacteria into into a bin for that specific bacteria, and then utilize traditional transcription analysis approaches to do species level transcription analysis within the microbiome during antimicrobial exposure. And here I'm just going to show you the data from B theta because it's an important bacteria that I'm discussing here. And what we found is that B theta, remember, is the bacteria that bloomed after amoxicillin exposure. We found that it seemed to upregulate a lot of enzymes responsible for fiber utilization, while at the same time knocking down the utilization of glucose as a carbon source. So a lot of simple sugar importers were actually downregulated, while the utilization of polysaccharides was upregulated during amoxicillin exposure. This was confirmed by qPCR. And B theta is in many ways, I like to think of it as a spider. So it does a lot of its digestion extracellularly. It digests long polysaccharides outside of the cell and then imports smaller subunits for fermentation. And we essentially found that the whole sus operon, this extracellular enzymatic complex, was upregulated in response to amoxicillin. So the question is, why is B theta doing this? And can we answer this in the context of now in vitro? Now, can we utilize the mouse data to motivate some in vitro experiments? And so essentially what we did is we took mineral media and we grew B theta in that mineral media, either with glucose or with dextrin, a polysaccharide, as the sole carbon source. And we found that if the bacteria is utilizing glucose, it's much more susceptible to amoxicillin than if it is utilizing dextrin. So in many ways, it looks like glucose is associated with death, but utilization of, fermentative utilization of dextrin seems to be associated with survival. We tried the same thing with the other two antibiotics, and we didn't find the same pattern. So this is only true for amoxicillin. Next, we also looked at to see, how would combining the two carbon sources work, and it appears that if you add dextrin to a mouse, to B theta that has glucose in it already, dextrin seems to provide some protection, but the utilization of glucose is still associated with a significant increase in susceptibility. Another important question is this just simply a growth, a growth rate dependent phenotype? And we looked at the doubling time on different carbon sources associated with and their MICs, and we found that actually the bacteria grows faster than dextrin, even though it is less susceptible to amoxicillin. So the doubling times don't seem to have an impact on whether you're going to be susceptible or tolerant to the antibiotic. So next, we kind of wanted to see, what is the kind of the metabolic signature that we have of this in a mouse. So what is the metabolic state that the mouse microbiome is encountering during treatment? So we did a total metabolomic analysis of the cecum. We found some really interesting things. One of them made me super happy. So this is data from a paper that I published in 2015 when I looked at the metabolome of E. coli treated with 3 different antibiotics, one of them being beta-lactam, in this case ampicillin. And we found that treatment with amoxicillin is also associated with significant changes in the levels of these of things like succinate. But the most dramatic impact that we found was the change in fecal glucose level, both through the metabolomics, but here we're doing kind of a direct quantitative analysis in the cecum of our mice. And we found that amoxicillin really reduces the concentration of glucose that's found in the cecum. Interestingly, ciprofloxacin does not have the same effect. And in fact, there's a non-significant increase in the levels of glucose in these mice. I'm not sure what accounts for this, but it tracks really closely to firmicutes. And firmicutes are likely important for the breakdown of polysaccharides into simple sugars in the context of the cecum. And that's probably where most of the glucose is coming from in the cecum, because mice are very efficient at absorbing glucose in their upper digestive system. And so most of the glucose found in the cecum likely comes from the activity of other bacteria. So what does that mean for susceptibility? Well, essentially, the non-antibiotic treated microbiome has a high fiber content and fairly normal glucose levels. Right, and so in this case, there's some glucose, some fiber, so it's kind of this intermediate response associated with susceptibility. However, when we give amoxicillin, we see that there's a collapse in glucose, but the fiber is still probably just as high, and this is more associated with the tolerant in vitro experiment, essentially associated with the exposure of B theta to amoxicillin on dextrin. So low glucose, high fiber seems to be also associated with protection in the context of the microbiome. So now let's kind of think about, like, what does this mean in the grand scheme of things? So typically, people eating our current fairly crappy diet, we would be given a probiotic to maybe protect our microbiome. Recent data indicates that's probably not beneficial. So this is one of my favorite papers where volunteers were given an antibiotic and then were either told to take a probiotic, do nothing, or do a self-fecal transplant. And what this group found is that essentially the probiotic reduces the capacity of the microbiome to recover. So it's actually detrimental. So maybe we can look at something else. Maybe we can feed the mice or humans a prebiotic, a fiber supplement that may protect the microbiome during antibiotic exposure. And a wonderful graduate student in the lab, Swathi, is actually looking at that right now. And so she is doing a set of experiments where we give mice different fiber sources at different times to look at how they impact antibiotic-induced microbiome disruption. In this case, she gave 2 different fibers to the mice before antibiotic exposure, and looked at the composition of the microbiome before, during, and during the recovery after the antibiotic, in this case amoxicillin. And what she found is that when we give just a single fiber, pectin doesn't really change the reduction in microbial diversity over time, but it does promote recovery after treatment. So these mice recovered their microbiome composition more rapidly, at least the diversity. She then did an experiment where she looked at fiber cocktails. So mixture of all fibers reminiscent of a normal mouse diet, given before, during, and after antibiotic treatment. So this would be associated, essentially a person living a very healthy lifestyle getting antibiotic. This is a person getting an antibiotic and fiber supplement together during treatment. And this is a person getting a fiber cocktail after they've already been treated. And we found that when we provide the antibiotic, either the fiber supplement, either before or during the antibiotic exposure, we have a less dramatic collapse in the diversity of the microbiome and a better recovery when we compare it to the control or the fiber that's given already after the antibiotic have been provided. And so this indicates that there's probably a really important role of carbon utilization in the susceptibility of the microbiome to antibiotics. So overall, kind of what I showed you today is that metagenomics is a really good way to look at antibiotic-induced dysbiosis, tells us who's there. Metatranscriptomics can tell us what are the pathways utilized by the microbiome, and that changing the capacity of the microbiome to utilize carbon changes the susceptibility of some bacteria to antibiotics. In the case of B theta, it activates fiber utilization. That appears to be protective. And then fiber is protective both in the microbiome and in vitro. And the main point is that changing our diet will change the susceptibility of the microbiome to antibiotics. And that's an important next step in my research. I'd like to thank members of my lab, collaborators that are both Brown and former collaborators in Collins' lab. We're looking for a postdoc, and I'd like to thank my funding sources. And again, I'd like to thank the organizer for this opportunity to speak to you in this recorded format. Thank you so much.
Hello, everyone. I am Dominique Lamoli. I'm an assistant professor at the University of Iowa. And I'd like to start by thanking Boo and George for organizing what I'm sure is going to be a really great session, even in these difficult times. And I'm grateful to have the opportunity to be a part of it and tell you a little bit about what my lab does. I have no relationships to disclose. So my lab studies polymicrobial infections in CF patients, and as such, I'm obligated to show you this slide from the Cystic Fibrosis Foundation, showing the percentage of individuals who are infected with different pathogens and that prevalence with age. And what you can see is that Pseudomonas and Staph are the two most prevalent pathogens in CF patients, and Staph is most common early in young patients and begins to decline, although less so than in previous years, as patients age. And in about the teens, Pseudomonas starts to become now the most prevalent pathogen. And so not only am I obligated to show you this slide, but it is also set the foundation for several of the hypotheses that we have in my lab, but then also in others. And one hypothesis is that Pseudomonas outcompetes Staph and replaces it as CF pulmonary disease progresses in patients. And so one of the ways that my lab studies Pseudomonas and Staph and co-infections and tries to answer this question is through a biofilm model on CFTR deficient bronchial epithelial cells, where we can grow a polarized monolayer of epithelial cells, and we can infect them with Staph aureus and let Staph grow, attach and grow and form microcolonies over time, and we can monitor the CFUs as these biofilms grow. If we let a biofilm grow of Staph for 24 hours, then we can infect with Pseudomonas and allow it to form microcolonies. And then we can follow their CFUs and ask whether or not Pseudomonas is able to replace staph in this model. And what you can see is that Pseudomonas grows very well with or without in monoculture or in co-culture with staph. But staph will decrease over time when we add Pseudomonas and as Pseudomonas begins to increase in abundance. And so this is very similar to what we see in CF patients. And so one might conclude that Pseudomonas can outcompete and replace staph as CF pulmonary disease progresses. However, we think maybe, and that's because there are some things that these data don't show when we look at the prevalence in all CF patients as opposed to in an individual. So these data do not show the longitudinal patterns of infection within an individual patient. So we really need to be able to follow patients over time in order to get an idea of what's happening with their infections. And we also need to be able to see the abundance of each species within an individual. So maybe there are, as a patient gets Pseudomonas, then the abundance of staph significantly decreases, even if it's not completely removed from that patient. So we wondered whether or not bacterial competition could be identified within individuals during infection. And so in collaboration with Tony Fisher and his lab at the University of Iowa, we asked whether or not we could identify CF patients in our hospitals that where we could actually follow them over time. And we were lucky to see that the clinical lab at the University of Iowa has actually been collecting these data for decades. And so we looked for patients who had sputum or bowel that had quantitative cultures. So this was such that the actual CFU per mil within these samples had been identified for both at least Pseudomonas and staph, and then also for other pathogens. And we asked that they had at least 10 quantitative cultures. And so with those qualifications, we found 134 patients that fit those criteria. And the median time that they had been followed was about 10 years. So this is an example of just one patient, and I'll orient you to how these data are presented. So on the x-axis, we have the year that the culture was taken. And if the patient had a culture taken at that time, there's a black dot. So anywhere that there's a gap here, there just was not a culture taken. And so that's why there's also a gap in the infections. And then on the y-axis, we have the quantitative culture. So how many CFU per mil on a log scale was cultured from an individual patient. And so we have Haemophilus in purple and Pseudomonas in green, and we have methicillin sensitive and methicillin resistant Staph aureus. What I hope you can see immediately from looking at this one patient is that these patients have multiple different organisms that they are cultured at very high CFUs, and 10 to the 8th is the highest that we were able to measure. So it could be even higher than that. But they keep these infections for a long period of time, and they have multiple different organisms. And here's just an example of another patient who has both methicillin resistant and methicillin sensitive Staph aureus and Pseudomonas. And you can see that all of these organisms coexist within a single patient at very high levels for a very long time. And here are just a couple of other examples of other patients. Here's one patient that had Pseudomonas first and then acquired a staph infection later. And then a different patient who had both methicillin sensitive and resistant staph first and then acquired a Pseudomonas infection. And so when we looked for patterns within these patients, what we really found was that there wasn't a pattern aside from that they could get different types of infections. They could get them in any order, so they could have staph first or Pseudomonas first. One was not more common than the other. And these infections were quite durable. They had them for many years at high levels, despite having very high, intense antibiotic treatments. Almost all of these patients had multiple different types of antibiotics, almost consistently. But the question that we really wanted to ask was, if a patient had a staph infection and then acquired a Pseudomonas infection, would Pseudomonas be able to replace staph within an individual patient? So we asked, with that first appearance of Pseudomonas, how long would it take until the last positive culture with that staph? So how long would it take for Pseudomonas to replace Staph aureus? And so here are the data, and again, I'll orient you. So on the y-axis, we again have the years to the last culture after Pseudomonas infection. And so immediate replacement of either Haemophilus or staph after a Pseudomonas infection would be around 0. And so you can see that it's only a couple of years on average until Haemophilus is replaced, but staph, both methicillin sensitive and methicillin resistant, it can take 5 years to even 10 years or 15 years after the appearance of Pseudomonas. So we really didn't see evidence to support our hypothesis that there would be replacement of Staph aureus after infection by Pseudomonas within an individual patient. And what we saw is that co-infection with Pseudomonas and staph was really common. It lasted for a really long time, and that the prevalence of Pseudomonas and staph co-infection would increase with age. And now if we look at the CF registry data, again, there are new data for the 2018 registry report showing that Pseudomonas and staph infections are indeed quite common, and they also increase with age. And you can see also dual infections on this report for multiple other organisms. So why do we care that Pseudomonas and staph infections are really common in patients? Does this influence outcomes for these patients to be co-infected with these organisms? And so we do see that co-infection correlates with poor health. And so studies from our studies, as well as multiple other groups in CF patients, have found that co-infection with Pseudomonas and staph increases pulmonary exacerbations, decreases lung function, shorter lifespan, and increased inflammation. And in animal models, both for airway infection as well as in wounds, that there's increased inflammation and delayed wound healing. So from these data, one question that really drives some of the basic science questions that we ask in my lab is whether or not bacterial interactions actually influence CF lung function, or if do Pseudomonas and staph actually interact with each other. And while I don't have an answer to that question, I can tell you a little bit about some of what we do know and some of the ways that we're investigating these interactions. So again, on our model of CF bronchial epithelial cells, here's some microscopy looking at what that looks like before when we infect Pseudomonas and staph on these monolayers. And what we constantly see is when we fluorescently label staph in red and Pseudomonas in green, is that Pseudomonas and staph form mixed aggregates on these epithelial cells. So they form these, they attach to the surface and then form little clusters together. And we wondered whether or not this would actually happen as well in CF patients. And so we've acquired sputum from co-infected patients and performed FISH on these samples. And we see indeed that also within samples from patients, we're seeing mixed aggregates of Pseudomonas and staph from patients. And so we wondered whether or not their Pseudomonas and staph just happen to be in the same place within the airway, and they happen to like to attach to the same place on epithelial cell. Is there something that actually brings Pseudomonas and staph together to form these microcolonies? And so we asked how these microcolonies might form, and one way that we've done this is by imaging the very early interactions between Pseudomonas and staph. And we wanted to image them on a single cell level in a way that we could continuously track individual cells over time. So we inoculate the cells between cover slip and an agarose pad to keep the cells in the same plane. And here's just an example of what Pseudomonas looks like on its own. So these are individual Pseudomonas cells that will replicate and divide outward. And what we see here and what others have seen is that on a surface, Pseudomonas tends to replicate and divide and move outward in groups with these tendril formations that are typical of type 4 pili-mediated motility. And so we then wanted to ask, could we image Pseudomonas and staph coming together and forming microcolonies on the surface? And so we added staph to this imaging, and here's a staph microcolony forming, and Pseudomonas, this is about 4 hours into this co-infection. And what you'll see is that as Pseudomonas begins to approach staph, instead of moving as a group, we see that individual cells break off from a Pseudomonas raft, and then they move with increased velocity and they surround the staph colony, and you'll see even more cells moving into the field of view and moving and surrounding this staph colony. And so from these data, it looked to us that Pseudomonas appeared to be able to sense staph from a distance and move towards it. And if we zoom in a little closer on when Pseudomonas very first approaches the staph colony, what we see is that they orient perpendicular to the staph surface, and the cells move, have increased reversals, and they move back and forth on the surface of the staph colony. And if we follow these cells, we actually see that the Pseudomonas will invade into the staph colony. And we can again label Pseudomonas and staph with different fluorescent proteins. And here we're able to see that Pseudomonas cells will move into the staph colony. And so a lot of what my lab is working on now is trying to understand this behavior. So we want to know how it is that Pseudomonas moves towards staph, and what it is that staph is secreting that Pseudomonas is able to respond to. I don't have time to tell you all about this today, but I'll tell you a little bit about what we know. So we know that even though Pseudomonas can move by flagella and type 4 pili-mediated motility, that this behavior is primarily driven by the type 4 pili. We can track individual Pseudomonas cells and track them as they move towards staph, and we see that the Pseudomonas cells move with a preferred direction towards the staph colonies, whereas when they're not present, they move in a more random direction. We also know that the signal that Pseudomonas can respond to, that staph secretes, are these small peptides called phenosoluble modulins. And when staph is unable to secrete these peptides, Pseudomonas again moves without a preferred directionality. So we think that they're necessary for Pseudomonas to sense and move towards staph. So if you are interested in learning more about what we're up to investigating these behaviors, I encourage you to check out posters from the graduate students in my lab. So Katie is poster 275, and she is studying how it is that Pseudomonas can sense gradients from staph and other organisms to move towards it, and what are the genetic and molecular mechanisms that drive this behavior. And Andrea is interested in what are the consequences of this behavior on Staph aureus. So we see that, again, Pseudomonas moves back and forth on the staph surface. So she's interested in how Pseudomonas is performing those reversals and what this means for staph and staph's physiology. So I'd like to thank the members of my lab who have made getting the lab set up and starting everything really exciting and fun. And I've had really incredible collaborators at the University of Iowa. So Tony and Sachin were instrumental in the quantitative culture experiments and the paper. And Tim Yar and Mike Welch have been really wonderful mentors for me with getting my lab set up. And George and Joanna were wonderful postdoc mentors that helped me initiate all of these studies. Ambrose Chung and Alex Horswell have been instrumental in teaching us and helping us with staph and giving us mutants and teaching us how to make mutants. And Diane, Will, and Peter taught us how to do the fish and BiPact. And I'd also like to acknowledge our funding from the CF Foundation. And I'm really excited to talk with you all later at the discussion sessions. And I hope to see you all in person very soon. I miss you all very much. Bye.